Hi, everybody. So it is my distinct pleasure to introduce Karen to you today. Karen has made this really easy on me because she let me know she's going to take us through a little bit of her journey as a young scientist in some of her talk. But I just want to say a few things on behalf of Karen and thank her for being here. You'll hear from Karen that she did undergraduate work at UCSD, PhD at the University of Oregon, and a postdoc at the University of Washington before landing an assistant professor position at the University of Minnesota in the Department of Entomology, and also an affiliation with the grad program in neuroscience there in 1988, and was promoted to full professor in 2001, which is an honor that she still holds. And her long-term service at University of Minnesota, which has become sort of a rarity these days in academics as well, with people shifting all over the place, I think speaks volumes about her dedication to not only her colleagues, but particularly to students. And I'm just going to talk about three quick aspects and get out of Karen's way so she can tell us about her beautiful work. Because I've known Karen actually, quite frankly, for probably longer than I've known any of you folks, even though I've been an STG person for a good long time. I started my life as a biogenic amine behavior person, and Karen was one of the first people I encountered in graduate school. And so I've known of her and known her for quite a while and know her good works. And so I just want to bring up a couple few things to make sure you know about them. Karen has been a inordinately prolific supporter of neuroethology across the world, most recently as the president, of course, of the International Society of Neuroethology, but has really used that platform to support young investigators and underrepresented scientists in ways that really speak to her mentorship of the community of neuroscience and neurobiologists and behavioral biologists in a way that I think we all know her to be an incredibly supportive and nurturing individual, but also a damn fine scientist in parallel with that as well. I also want to point out that she has been, in addition to helping young scientists, really at the forefront of making sure that women in science are represented and have every opportunity to demonstrate the incredible contributions across the board. In particular, you should know that Karen was awarded the Ada Comstock Distinguished Women's Scholar at the University of Minnesota. If you don't know who Ada Comstock is, she was really a pioneer in women's education, particularly higher education. She was the first dean of women at the University of Minnesota and then went on to serve as the president of Radcliffe College. And thanks to the wonders of Wikipedia, I just want to say that ultimately what is said of her is that of Ada is that in her capacity in these positions, she was instrumental in improving the quality of life for women of the college, arguing persistently that a college was responsible for one's physical and intellectual well-being. I cannot think of a better person to represent the legacy of Ada Comstock than Karen, because not only does that fit her to a T, improving the quality of life, the quality of opportunity for women and all of us in science, but that this is inextricably linked with our physical, intellectual, and mental well-being, because she's an incredibly supportive and nurturing mentor. And that goes along with a dedication to students with a Distinguished Teacher Award from the University of Minnesota as well. And then finally, I have to inject the personal anecdote as the third part, because I want to say back in 1997, Karen organized the Midwest Neurobiology Conference, affectionately referred to as the No Coast Nerve Net at the time. That was one of the very first meetings I had ever gone to and presented at. And I, like most graduate students starting out, was incredibly nervous. Felt like the folks whose papers I'd been reading for those past few years were this elite class of individuals that I dare not sully their shadow. And Karen was one of the first people at that meeting to come up to me and make sure that I felt totally comfortable, make sure that I felt like I was a colleague and a peer at that meeting. We had met each other earlier, so we knew one another, but I felt like in that moment, not only did I experience Karen as a scientist, because I do remember, Karen, you asked me one of the most, I think, cutting questions after my talk possible in a good way, right? That really drove the experiments further. But as a mentor and somebody who cares about students deeply, I got to experience that firsthand. And so finally, the last thing I will say is I am now actively engaged in a collaboration with Karen, which I feel is one of the pinnacles of my scientific career. You start out and you read papers from folks and individuals, you develop sort of this in absentia respect and admiration for them. You meet them and they turn out to be even better than you could have imagined as individuals. And now 20 plus years later, you get to work closely with them. I consider this one of the greatest privileges of my scientific career, Karen. I really enjoy working with you and I've always loved your work. And then the last thing I'll say, just as a side note, in your biosketch you sent me, you credit yourself with the Adam Comstock Distinguished Award. I don't think Adam could hold a candle to what Ada has done or what you have done. So whoever that guy is, he needs to be gotten rid of from your biosketch to make sure that everybody knows exactly whose legacy you're carrying on so ably. So thank you for letting me introduce Karen. Karen, thank you for letting me take a few minutes of your precious time here. I'm going to get out of the way. I can't wait to hear what you're going to talk about today.
Wow, thank you so much, Dave, for that kind introduction. I'm blushing, actually, but it's mutual. I'm enjoying our collaboration, so I want to thank you again. And with that, I will share my screen and hopefully I'm going to move some things out of the way. And I'm going to click on this. Everybody can see this just fine? Okay, great. Wow, it is really a pleasure to be able to share with you the work that I've been doing. It's really great to talk to an audience that has a deep appreciation for a simpler systems approach. So I'll be talking from shells to cells. I'm not going to say that 3 times fast. And I'm basically going to share with you my journey to understanding decision-making and action selection in simpler animal systems. And here we have a picture of the Minnesota Biological Field Station up at Lake Itasca, which is the headwaters of the mighty Mississippi River. So I was born and raised in Jersey, so I am truly a Jersey girl. But my dream is to become a marine biologist. And so I headed out to California. To the dismay of my parents, they broke their hearts when I went that far away. And I attended UCSD. This was really the beginning of my scientific career. And I worked with some wonderful people. I went down the hill from UCSD and worked with Ted Bullock for a little bit. We had some scheduling conflicts, so I was unable to go with him to Marine World and study cetacean acoustical behavior. And I think back, wow, if I didn't have to take that PChem course, I'd be in some tropical islands, swimming with the dolphins. But nevertheless, Ted introduced me to Walter Heiligenberg, who was a newbie to Scripps Institute of Oceanography. And I worked with Walter for several years, and I'll talk briefly about the work I did with Walter. And that, last but not least, was Dan Hartline. I was a TA for Dan, and I had a little desk in his lab, and while I'd be grading quizzes, I would hear out of the audio monitor this pulsating sound. And that's when I was first introduced to the STG by way of that pyloric rhythm through his audio monitor. So I'd be grading quizzes to the beat of the STG. All right, so in Walter's lab, I studied hermit crabs. Walter knew that I had a penchant for marine organisms. And so he had an idea about me looking at the role of social isolation in inducing aggression. And it was a project that Walter had with Conrad Lorenz. And I'm not quite sure why Walter even got engaged in that particular project, but he gave it over to me. And I didn't work on electric fish with Walter. I worked on hermit crabs. And I found that really social isolation was not inducing aggressive or agonistic behavior, but rather it was the isolation. Once these hermit crabs were no longer isolated, they wanted to exchange shells with the other hermit crabs. And so they expressed something called knock-rock behavior, where one crab would basically embrace the other and would bang the heck out of its shell on the other shell and eventually evict the hermit crab from its shell. And sometimes these dance partners would be better off because they had better shells, and sometimes one was left with a crummy shell. But all of this led me to the curiosity of how the heck do hermit crabs select a particular shell? They're very, very picky, right? If they're given maybe 100 shells, they'll just basically look through all of them until they find their ideal shell. So I was very curious about how they made a decision. How did they weigh all these variables about size and weight and texture and color, the integrity of the shell? How did they do that? So that was really what was driving my scientific pursuit of studying of hermit crabs. Anyway, of course, I couldn't finish these studies while I was at UCSD. So I went off to the University of Oregon for grad school, and I made a home in the lab of Graham Hoyle. And here we can see Graham as a younger chap here. And here's a photo of him as most of us remember him. And I have to say that I learned a lot about Graham. He would have tea between 4 and 4:30 every day, and he would tell us stories about the Huxleys and other things. But I learned a lot from him. And I have to say, he was very generous. He gave me free rein in the lab, and he was never mean to me. I know that he was pugnacious was a bit nasty to some other folks, but he was never that way to me. And what I want to note is that Graham had a number of scientific children, including Al Selverson, Dennis Willows, Jack Davis. And so these were my scientific brothers, which makes me a scientific auntie and great auntie to a number of STG researchers. Okay, so when I was in Graham's lab, I was very interested in decision-making processes, and I was interested in decision-making long before it was fashionable to think about decision-making and the neural mechanisms. So essentially, my idea was, let's make a replica shell. Here's a natural shell. This was selected by the hermit crab from over 100 plus shells. And my idea was, if I made a replica shell, then I could independently vary the size or shape or weight of the shell. And so my initial experiments were, I wanted my replica shells to basically not be rejected. And I was hoping for about a 50-50 choice between the natural shell and the fake shell. And inevitably, the hermit crab kept selecting my fake replica shell. Now, I conducted these experiments in darkness, so color wasn't a factor. And I was really frustrated because as a shell architect and carpenter, I needed to make shells that were really a full replica. And this is a shout out to graduate students who get frustrated and are about to abandon a project, which I almost did, but I made lemonade out of lemons. And actually that led to my first publication, which was in Science. And what I found was that it was the chemical makeup of the plaster of Paris that was very productive in a sense. Calcium, natural shells are made of calcium carbonate, and plaster of Paris has calcium sulfate, which basically, exudes more calcium. And I found that even if I made a blob of plaster of Paris, the hermit crab, the hairy hermit crab in particular, with this little soft curled abdomen, would select the plaster of Paris blob over the natural shell. So in a very ethological way, I was able to identify a sign stimulus, in particular, super sign stimulus. And that was a way of feature extraction. And this particular cue I found was able to elicit a sequence of stereotypical shell investigatory behaviors. And so once this hairy hermit would be rolling the shell and exploring it with its minor and major chelipeds, it would be convinced it was a shell. And even though it didn't have an aperture of a shell and it didn't have any opening, it would pivot and try to basically insert its soft abdomen into this blob of calcium. And it would repeat this over and over again to the point where its little abdomen was bruised. But that's about as far as I could get in trying to understand decision-making. I was stuck at the sensory level, eventually identified the chemoreceptors, and I could see where they projected. But to understand how action patterns are selected at the neural level, and to understand how specific sequences of behaviors are controlled, these are tough nuts to crack. And the hermit crab, although a wonderful animal system to study decision-making, it wasn't amenable to neurophysiological analysis at that time. Well, when I was a graduate student, I'd heard Jim Truman at the University of Washington talking about how sequences of behavior could be dictated by the sequential release of hormones. And so I was very fortunate to be accepted into Jim's lab at the University of Washington. Here we see the campus with Mount Rainier. Jim's favorite preparation was the hawk moth, shown here in the adult form and in its larval form. And I always laugh when I see this picture because I had been talking Jim's ear off to the point where he just became dazed and confused and nearly comatose. And here he is, very stunned looking. But I have to say, Jim is one of the most accomplished scientists I've ever had the pleasure to work with, and we're still very close colleagues, and I admire so much of his work. So in Jim's lab, we studied ecdysis behavior, and ecdysis is a universal arthropod behavior. It's a precise sequence of behaviors that removes the exoskeleton from the animal. We know it's centrally driven. It lasts between about 10 minutes to an hour based on the particular species under investigation. And I have to tell you, it's probably one of the most universally expressed behaviors on the planet if you think about how many species on our planet are arthropods. And they can't make mistakes. So it's really important that here we have a grasshopper, that this animal basically get all of its little bitty parts off. And it has this stereotypical sequence that it uses so that it doesn't get stuck and die. And in Jim's lab, we looked at a number of neuropeptides that would be released in a sequence to give rise to particular behaviors. So for example, we have these hormones inducing a pre-ecdysis behavior 1, a pre-ecdysis 2 behavior, which help loosen the cuticle. And then we go into full-blown ecdysis behavior, and then there's a wing expansion, cuticular tanning of the cuticle, et cetera. And I worked on these various neurohormones, and I focused in on adult-specific ecdysis behavior. Now, mind you, when an animal molts from one stage to another, like a caterpillar molts to another caterpillar, it has a relatively simple behavior. It expresses a relatively simple motor program. But when the animal is at a pupal stage and it's molting to an adult, it has a very more complicated program because the animal is underground in its underground pupation chamber and has to not only get rid of its exoskeleton, but it has to dig itself out of the ground. So caterpillars have this sort of metachronal wave that goes posterior to anterior direction. And these little bars are reflecting the muscle activity that I would record from with little wires. And if I were to record from the same muscles, but in an adult, I got this very rapid peristaltic wave with wing shrugging, and then this coactivity of musculature that cause the abdomen to be propelled out. And so in essence, this is more like a digging behavior with a retraction and then an extension of the abdomen. And so Jim put me on the most difficult project in the world, trying to find the CPG for ecdysis. And I have to say that to my knowledge, no one on the planet has yet identified an ecdysis central pattern generator. So what I was able to do in Jim's lab was start to pull apart the nervous system. I took away the brain, SEG, some of the thoracic ganglia, and with the neuropeptide, I would get full-blown adult-specific ecdysis behavior. But when I took this region of the nervous system away, in pursuit of this, central pattern generator, I found something really interesting. And what I found is that the adult no longer behaved like adult. It expressed a larval-like motor program. So here's a motor program that is now being expressed by the adult. And through lots of other studies I don't have time to present, we found that this motor program expressed by the adult was indistinguishable from that expressed from the larvae. So this was really inspired a lot by the work that was done on the STG, thinking about polymorphic networks, and maybe there was some kind of descending influence that basically, changed the output of some retained larval ecdysis circuitry. Later on, I found out that there were two neurons in particular that seemed to be responsible for this modulation. It was very difficult to do neurobiological experiments in Manduka. They have a very thick sheath. You can't see the cells, so it's problematic. Okay, the next slide, I'm going to show you what I did was I wanted to reversibly block the descending information from this pterothoracic ganglion with this kind of a la STG like understanding that these modulatory projection neurons could maybe change the output of the conserved larval CPG. So I couldn't use the sucrose block because of all the sheath problems in the insect. So I built a little mini refrigeration unit and I applied this cold block right here. And when I applied the cold block and I blocked descending input, then I unmasked this larva-like metachronal wave, indistinguishable from what you would see in a larva. When I turned the cold block off, enabling descending information to prevail, within about a cycle or so, I would then see the adult-specific motor rhythm. And I could turn the cold block on and off and on and off and keep switching between the larva-like and the adult-specific pattern. And so the take-home message here is that with polymorphic networks and this introduction of these projection neurons, you can create a new motor program. And this was being done during development. So it's a nice trick during development to use your old circuitry and modulate it for a novel behavior. Pardon?
Okay, have it your way.
Their question?
Oh, sorry, it wasn't muted. Talking to my experiment.
Okay, no problem. Okay, all right. So I was influenced by Walter, who introduced me the idea of champion animals, and champion animals were animals that could give you a solution to a biological problem more accessibly than other organisms. And this leads me to the wonderful leech. I've been now working on the leech for a number of decades, and I was introduced to this preparation up at Lake Itasca, where we had about a 6-week-long NSMB-like course, and we would basically have all of our graduate students in our neuroscience program go up to Lake Itasca and learn about the leech. And I was invited up there when I first became a new professor at University of Minnesota, because Ron had been invited previously to give a leech a week-long leech tutorial on how this wonderful system works. So I really owe it all to Ron. And then my graduate student who was with me at the time, Laura Gilchrist, she was like, I'm not working on Manduka anymore. I want to work on leeches. They're really cool. And it was really great to work on more accessible neurons. So the leech is indeed a wonderful preparation. Some of you know because you learned about the leech if you were in the NSMB course. They have large and identifiable neurons, and they're so pretty. They actually graced the Journal of Neuroscience for us one year. And they have about 200 pairs of neurons. They have a cephalic ganglion, so they're definitely capable of having decision-making and executive functions with that. They have 21 of these wonderful, glorious ganglia. So if you make a mistake with one, you can just pluck out another one, so they're very cost effective. And especially for me, they lack a formidable blood-brain barrier, which means it's easier to conduct pharmacological studies and also conduct immunocytochemical projects are better, because the antibodies have a better chance of penetrating the nervous tissue. And just by the way, in 2018, we identified the stomatogastric ganglion for the first time in leeches. And that was, we got the cover there, little rockets of leeches showing their biting. And the STG, there's actually about 5 ganglia that are involved in the biting and feeding aspects and swallowing. But that's a story for another day. So when I began focusing on the leech, I gravitated towards crawling because it was less known about crawling behavior in contrast to swimming behavior. Otto Friesen and Bill Christan had done a wonderful job identifying the swim central pattern generators, and but very little was known about crawling. Okay, so if you don't know, this is the front of the leech, this is the back of the leech, and the behavior is relatively simple. They elongate their bodies, plant their suckers, and then they contract, release a sucker, and this is continued over and over again. So there's an elongation phase followed by a contraction phase. And what's really important for you guys to note is that this peristaltic or metachronal wave always goes from the front to the rear direction in a very precise sequence. So this anterior to posteriorly directed wave occurs during the elongation phase and during the contraction phase, where in this case, each segment will contract in an orderly fashion front to back. That is really important to remember because I will be coming back to this crawl-specific intersegmental coordination. Focusing on locomotion is fun because you can look at action selection. You can ask, how is one form of locomotion, swimming or crawling, selected over another? And you're able to have a lot of problems that you can, solve, lots of neural solutions to these problems. You can look at the initiation of the behavior, the rhythm generation, shifting gaits, stopping the rhythm, adaptation to the environment. And one of my favorite aspects of locomotion is coordination. Basically, how do you get appendages to move in some kind of coordinated way, left to right, front, back, one particular appendage, etc. All right, I'm going to cut to the chase here and tell you that we discovered that dopamine is a really potent activator for crawling, plays a really major role in action selection. In parallel with this ability to activate crawling, it is a potent suppressor of swimming. So in the presence of dopamine, you cannot get swimming. And I was at NSMB with Bill Christan one time and we made a bet. He was like, no, I can get it to work. I can get it to work. And he lost. So I won that bet. So you really can't, even with electrical stimulation, what have you. In the presence of dopamine, no swimming. And this work was conducted by my graduate student, Josh Poole. He went off to NIH eventually to work with Michael O'Donovan, and then he missed the leach and he came back and did some additional work with me as a postdoc. All right, so right off the bat, we know that dopamine is really critical in shifting or biasing the nervous system to express crawling. Well, we went to the fictive preparation, of course, and I just want to briefly talk about some of the key criteria that are needed to convince one that indeed you are looking at a fictive crawling rhythm. So there's two motor neurons I want you to focus on, the circular motor neurons and the longitudinal motor neurons. Here's oscillations in the circular motor neurons, and they innervate circular muscles that basically when contracted, they squeeze in on the body of the leech, kind of like you squeezing a tube of toothpaste and then you wind up extruding the toothpaste. So activation of the circular motor neurons and the muscles associated with them cause an elongation of each segment. In turn, DE3, those are the dorsal exciters for longitudinal muscles. And those longitudinal muscles span each segment. So when they're active, a particular segment will get shorter. And lucky for us, the spike of DE3 is the largest unit in this nerve, which we call the DP, the dorsal posterior nerve. So we have elongation, okay, and contraction out of phase. And there are other criteria that we have to abide by. The dorsal and ventral motor neurons burst in phase. Bursting has to propagate along the nerve cord in an anterior to posterior direction. And the periods can't be too short or too long. They're typically between around 5 and 25 seconds. So in search for the crawl center pattern generators, just like I was in Jim's lab trying to get at regions of the nervous system, Josh basically was applying dopamine. Now we're using it as a tool to see if he could turn on a particular central pattern generator and what was the smallest unit of the nervous system in which he could elicit a bona fide crawl rhythm. And it turns out that when you apply dopamine to a single ganglion, you can get all the elements needed for crawling, all right? And I'm not showing you all of the neurons we've recorded from, but suffice it to say that each and every ganglion has its own central pattern generator for crawling. And then this will beg the question later on, well, how do these independent CPGs become coordinated across the nerve cord? And then here's just a recording of DE3 activity in an anterior ganglion and a posterior ganglion. So this was really neat because this became our unit burst generator, a la Sten Grillner like to call these things unit burst generators. And before I leave the dopamine story, let me just tell you that we didn't throw dopamine on willy-nilly. We've been working on a story about the development of the dopaminergic neurons. And here you can see them with labeling to tyrosine hydroxylase. Here's what I'm going to call this whole thing the brain, the cephalic ganglion, the terminal ganglion, and these mid-body ganglia. And this has really what got us interested in the role of dopamine. It started with Kevin Crisp, a graduate student in my lab, and Kevin was looking at this interneuron in the brain, TR2. It had been first identified by Peter Brodfuhr, and this neuron, TR2, was shown to inhibit swimming. So if we were to inject neurobiotin, we did inject neurobiotin into this neuron and filled it. We always found that these lateral cells also filled with neurobiotin. So the lateral cells were dicoupled to TR2, and these lateral cells happen to be the dopaminergic cells. And with double labeling, you can see the co-localization of dopamine immunolabeling with the neurobiotin labeling. And here's just what a dopaminergic neuron looks like in the brain. So just keep this in your mind. When TR2 is activated, it's suppressing swimming, and at the same time, it's electrically exciting the dopamine cells, which are sort of acting like a double whammy, because that also is going to suppress swimming. And Kevin also was then asking, well, when dopamine is released, does it do something more than just inhibit swimming? Might it turn on another motor program? And we were interested in whether dopamine could maybe help to switch from swimming to crawling, but Kevin was always getting crawling all the time, so we could never really show that that dopamine was inducing crawling. And that's where Josh came along, because in his hands, he would have a prep that was silent, and then we could turn dopamine on. I mean, we could turn crawling on with dopamine. So before I leave the story of dopamine, and I want to go back to this idea of action selection, we wanted to know about the role of the brain in making decisions about the form of locomotion to express, and also what its role was in locomotion in general. And for this, we wanted to observe the overt behavior of the body. And so here's a preparation that was developed by Teresa Esch, and it's the nearly intact preparation. And so we basically have isolated the nervous system here. We provide it with a bow tie, and in that bow tie, we can present the sucrose, isotonic solution of sucrose that can block descending information. And then in this chamber, we can vary the fluid levels. So we can create an aquatic environment or a terrestrial environment. And then we had 3 phases of study: a pre-block, a block, and post-block condition. And this is what crawling looks like in the nearly intact when it's in a terrestrial terrain. And then we looked at behaviors, spontaneous behaviors in high and low water. In high water, they typically will swim. Low water, they're going to crawl, and elicited behaviors. So here we have an ability to basically let the animal tell us what it's going to do when it doesn't have descending information. And we were able to test whether the brain was necessary and sufficient for overt crawling. So just focus in blue here, because that's the amount of crawling in low water versus green in high water. And you can see spontaneously they will crawl in pre-block and post-block conditions, but during the block, there was no spontaneous activity. We'll switch over to, well, maybe they weren't motivated enough to crawl. So in these conditions, we elicited crawling by poking their butt really hard with a stick. And 100% of the animals, 100% of the time, they would crawl. And they did that in the post-block conditions, but 0 of them would crawl, no matter how much stimulation we provided. We were a little evil to the leeches, have to say. I have to apologize to them. But I want to contrast this blocked condition with swimming. In high water, in the block condition, we have lots and lots of swimming. And that's because the swim CPGs are under the inhibitory control of the brain. So when we cut off the brain or reversibly remove the brain, these guys rock and roll with swimming. So real difference in the control of the system. So here we have a leech that is swimming and crawling. What we've done here is we're showing the role of the cephalic input to this portion of the nervous system. And here with a nice big bold transection, this part of the nervous system is not under the control of the brain. So cephalic dependency, cephalic independency. So you can see the metacronal wave, especially going in the during the contraction phase. And here is the posterior region swimming, because it's an aquatic environment. If the whole leech were basically further over, it would basically only be crawling at this end and dragging its butt end. All right. So at this point, it'd be like, wow, wouldn't it be nice to find a single neuron that can control crawling? And to cut to the chase, yes, we did. We found this neuron called R3B1. It is a decision-making neuron for locomotion. It's such a beautiful cell that Andy Warhol decided to paint it. Now that part I'm just kidding about. But anyway, we have a nearly intact preparation. We can go in and we can look for cells. And this is some of the work that I did when I was on a sabbatical with Bill Kristen, and then never forgot my first love of R3B1 and have continued to work with R3B1. When I joined Bill's lab and worked with Teresa, she had identified R3B1, actually a little combination with me, with a previous study that we were doing long distance backfills, basically placing dye and looking at what cells might go all the way up to the brain. And so she got a head start before I got out to San Diego. And so we have the body of the leech in the chamber. And we also have a fictive component because we can record the activity of identified motor neurons from that nerve. And here we are with the electrode stimulating R3B1. And Teresa was really frustrated the time I got out to UCSD because sometimes she would stimulate R3B1 and get crawling, and other times she got swimming. She didn't know if there were subsets of R3B1 like cells. And then spring break, everybody left the lab and I was all by myself, and I had this pet theory that I tested, which is I raised the fluid level of the chamber. And I thought to myself, if I stimulate R3B1 and I lower the fluid level so it's a terrestrial train, will I get crawling? Yes, indeed, I did. And so I was holding my breath and I raised the fluid level, still had the same R3B1 cell, and I no longer got crawling, I got swimming. So the decision is made by R3B1 is going to locomote, but the form of locomotion, crawling versus swimming, is shaped by the context in terms of that body being in a terrain that's aquatic or terrestrial. And then I was a little bit mean to the leech. I made it think a little bit. I'm being anthropomorphic here, but I had an intermediate fluid level. And what was really curious is I had swimming and crawl time. So I had a hybrid rhythm, a la pyloric, gastric, cardiac, pattern generators fusing and making a brand new behavior. And so we had this, this swimming, which is much faster rhythm than crawling, but over crawl time. So it would elongate while it was swimming, and it would stop and it would contract, and then elongate and contract. And those variables were more in crawl time. Okay, R3B1, it's called R3B1 because it sits in this third neuromer of the subesophageal ganglion in the B subpacket. Okay, and Teresa and I first were identifying these cells because we put the label down here and then watched for cells that were able to project that long a distance. And there's really only several dozen of cells in the entire brain that do that. And there's a pair of them. I'm showing you only one of the homologs. Okay, so what does R3B1 look like in the whole nervous system in a dish? It looks like this. It bursts. And here we have DE3 activity in the mid-body segments. And R3B1 likes to burst during the elongation phase. It's inhibited during the contraction phase. But what we really wanted to get at is how important is R3B1 to the whole system? We needed to silence R3B1 and ask the question, will crawl-specific activity be eliminated? And this was a really wonderful experiment that Josh did in my lab, where he has the whole nerve cord out. He wants to look at the effect of just a single R3B1, so he clips the hemiconnective here, preventing the contralateral homologue from having any effect on the nervous system. And he's recording and stimulating from R3B1 and then having the other segments monitored via DE3 activity in the DP nerve. In this experiment, we provided a sub-threshold level of dopamine, so we see these little ratty oscillations in R3B1. But when we stimulate R3B1 with current, then we get this beautiful crawl-specific metachronal wave going anterior to posterior, and we have our crawl periods sort of tighten up. And then this was the best experiment. So we have nice, fictive, bona fide crawling. When we inhibit just the single R3B1 cell, the crawl-specific metachrony goes away. We start losing isochronicity, and everything falls apart. Now, this is biased by dopamine, so we're still going to see some bursting in the independent crawl oscillators, but they're not coordinated intersegmentally. Right, so R3B1 is doing a lot. It's initiating crawling, and it also seems to be really important for the crawl-specific intersegmental coordination. But I don't want to leave you with the idea that that's the only thing that's playing a role in intersegmental coordination. And so here's some other experiments we did. We posed the question, is there some kind of oscillator to oscillator coupling? We wanted to activate one crawl CPG with dopamine, as we did here. Basically, this ganglion is in dopamine, and this is its activity. This anterior adjacent and posterior adjacent ganglia are not exposed to dopamine, but with just this CPG turned on by dopamine, it drove crawl-like bursting in the anterior ganglion here and the posterior ganglion here. And what was really cool is that the periods of the bursts were all the same, as shown here. And then we also found that the number of spikes in the posterior burst were always greater. So that's shown right here. All right. So here's our working intersegmental coordination model. The crawling is when excuse me, when in a crawling mode, okay, R3B1 is shown in this lime green. It provides a global activation signal to all the segmental oscillators shown here. And we think it permits the propagation of concatenated inter-oscillator signaling. So without R3B1, the individual oscillators are not going to drive each other. And then hypothetically, we just think there's some kind of logical AND gate that helps to set the rostrocaudal timing of the system. Okay, so that's our working hypothesis. What would happen if in the whole animal you somehow took the brain out or you perturb the system? You know, what would happen? And this is the rest of the talk. I want to talk about something that I find really amazing. And that's over time, if the crawl system is perturbed by removing descending influences, you know, like what's going to happen? I'm thinking homeostatic plasticity, but it really all boiled down to just an accident. We had a number of animals that we removed the brain and they could swim like crazy, but they never crawled. And we were showing a new student how without the brain, these animals couldn't crawl and they were crawling. And we were really confused and embarrassed. And it led us to a wonderful story. And that is that, yes, in the beginning, when you remove all this very important descending input, by around 2 weeks plus, they rock and roll and they show beautiful crawl-specific intersegmental coordination. they're motivated to crawl, and they will do that. And we don't have time for me to show you all of the data, just to show you that this is the timing of when things turn on, their crawlability. They're by about 10 days, they're able to crawl. It gets better and better over time. And what's intriguing is this excessive swimming actually goes back to baseline.
Hey, Karen.
Question?
Karen, can I ask you a question?
Yes,
Are these full transections or hemisections?
these are full transections. And actually, I want to point out here that we were wondering, well, maybe R3B1 and other descending cells are just growing back.
Mm-hmm.
So we've done experiments where we've actually taken out these ganglia in the middle, and we've done experiments where we've actually removed the entire brain. And through lots and lots of different experiments, we see no regrowth from the anterior part of the nervous system. So beyond just a hemitransection, these are full-blown transections, getting rid of the brain and getting rid of the intervening ganglia. All right?
And the swimming looks normal or?
Yes, the swimming looks normal. The swimming always had looked normal. It was just that it was not under any inhibitory control. So, you know, they were swimming to exhaustion almost until about 10 days later, they had a normal regulation of swimming. And that's a story, Jorge, that we would like to pursue at some point, because it's another layer to this crawl recovery. All right, so in the time I have left, which is not a lot, we had this idea that the anteriormost ganglion below the transection, you can see there's no growth here between the brain and M1. We had this idea that maybe it's remodeling and becoming a lead ganglion. And when we say leading the propagation of the wave in the anterior to posterior direction. So we had this hypothesis that if we had recovered animals that had a transection, and here they are fully recovered, if we then made a second transection, removing the lead ganglion, would that perturb their ability to crawl? And yes, indeed. So if you make a second transection, or you remove this lead ganglion, they have to quote unquote learn how to crawl all over again. They basically start from ground zero, and then it actually can take a little longer than the first transection, but the recovery comes back. So this gave us this idea that something special is happening within this lead ganglion. And here's a preparation in which we did the first transection, and then we actually did another transection where we removed the terminal ganglion, which controls the sucker. And then we did the removal of the lead ganglion, and that was at 81 days. And now we're looking at an animal 16 days after this lead ganglion was removed. They're a little wobbly because it's just 16 days, but you can still see, especially during the contraction phase, the metachrony. And what's amazing is they still have use of their suckers, although the sucker is controlled by what we call the head brain, and the sucker is controlled by the rear brain. And we know that there's no ingrowth or regeneration of processes through the connective. So they're really resilient and fascinating. So I want to just summarize this part of the talk and then take about 5 minutes to talk about how this is occurring and what we know so far. So, you know, just the global summary of crawling motor control in the leech is the following. Dopamine is a potent activator of crawling and potent suppressor of swimming. Under normal conditions, the brain is both necessary and sufficient for both overt and fictive crawling. This is critical. We know this for sure. A single R3B1 neuron is necessary and sufficient for coordinated crawling, but it's in combination with this oscillator to oscillator drive. By 2 weeks after removal of cephalic projections, leeches show robust crawl-specific coordinated crawling, and excessive swimming returns to control levels. And after successive transections, you can do this multiple times. Leeches show first a loss of crawling, but regain their ability to crawl. How is this working? Like, what is going on? So we had this hypothesis that maybe just like swimming, there's a rearrangement of the oscillators so they can just drive each other in a concatenated fashion. And we have shown additional drive from oscillators that go beyond their adjacent oscillator. So you would argue if we had a transected animal that recovered, we could put it in the dish, throw on dopamine, and we get beautiful fictive crawling. And that's not what ever happens. This is the motor output from an animal that has its brain. You can see the metachronal wave, etc. So we did all kinds of combination of chains of ganglia in dopamine, etc. We even had this idea that when we took a chain out from a fully recovered animal and we placed dopamine just on the lead ganglion, everything would fall into place, and it's not true. We get the bursting in subsequent ganglia, but we never see the crawl-specific metachrony. We even see phase advances, but we don't, the ganglia are gonna burst, but we never see what we wanted was the crawl-specific coordination. And I'm gonna move on here, but you can read our 2018 paper in eNeuro, and you can see we tried all different kinds of combinations, and we compared the non-transected controls and ones that were recovered, and we never saw anything good. Which leads me, I'm just going to take a break. I started with a cold last night. So I have a raspy voice. I want you to focus on green. We had this idea that, okay, we have beautiful coordinated crawling in the whole animal, that the whole animal is telling us that. But when we take out the nervous system, put it in the dish, put on dopamine, everything is just falling apart. We don't have any coordination, which says that afferent information is crucial for the generation of the crawl-specific coordination. So here's the experiment we did. Here's our lead ganglion. We denervated the lead ganglion. We had the transection, and we asked, you're looking green, whether those animals can recover. And some of them recovered, but not many. Only about a third recovered. And this is in contrast to denervating the ganglion below the lead ganglion. That's in purple, okay? They fully recover. And for controls, we had the animal in red, fully recovery, because we never transected them and removed the descending input. We just denervated them. And they didn't care. Matter of fact, they didn't miss a beat, okay? After that surgery, no detriment in the quality of the crawling. And then here in blue is just a regular old transected. You can transect here or here, it doesn't matter. They'll recover their crawling. And in blue, there's not as much recovery because sometimes it takes a little longer than 14 to 16 days. So what's going on? Here's the key question. Why do we have a decrease in the amount of recovery? And then the other question, the flip side, is why is the recovery at all when we denervated this lead ganglion? And here's the punch line here. That is, in recovered leeches, when we made the cut to these nerves, these afferents grew back into the ganglia. And in the unrecovered leeches, there was a scar that was developed, and these afferents could not make their way and pathfind their way into the ganglion. Okay, this is really cool. we have a lead ganglion. We know there's an importance of sensory information. So who are the cells that are now substituting for the timing of R3B1? And long story short, Susanna Blackshaw had identified the proprioceptors or the stretch receptors in the body wall of the leech. And John Jellies actually many years ago had looked at the development of these cells. They're known as Hoover cells. There's 8 of them in each hemisegment. And they're really sensitive. You don't see the little fibers coming off their cell body here. They're really sensitive to stretch of the body, which is what I think you might need to help control your coordination if you don't have R3B any longer. I can't stress enough that if you fill these stretch receptors, the terminals of these stretch receptors never, ever, ever, ever leave their home ganglion. And this was shown by Otto Friesen's lab also, because Otto had found that these stretch receptors played a role in swimming. And this is what's really exciting for us, is that recovered leeches, when we fill these afferent proprioceptors, they've become anatomically remodeled. They shoot out an anterior going process and a posterior process. Here's where we filled multiple afferents, and you can see these anterior and posteriorly directed processes, which is something you would need if you are going to be responsible for some kind of intersegmental coordination. Here's the control terminal of a particular stretch receptor, and here you can see one of the mistakes that it's made in its outgrowth. So we've looked at other changes in different mechanosensitive cells, the touch pressure sensitive cells, and see absolutely no change in their morphology. And then briefly, what about changes at the level of gene expression? And this is where my wonderful collaboration with Dave Schultz has come into the limelight. Is I'm just going to pick out just some of the preliminary data that we have. Of course, I was interested in dopamine changes in dopamine receptors, because we know dopamine plays a powerful role in initiating crawl activity. So these boxes here represent the individual ganglia, and this dotted line indicates where the transection was made between these 2 ganglia. We just call them ganglion 2 and 3. And at a minimum, Dave and his group have looked at changes in gene expression related to motility proteins, dopamine receptors, serotonin receptors, and synthetic enzymes for the biogenic amines. And here are some changes, and in this one in particular to the D3 dopamine receptor. This is a log2 fold plot. There is an 8-fold increase in the production or the gene expression compared to a sham ganglion, and a downregulation of D3 dopamine receptor expression in the ganglion that was just anterior to the cut. So both ganglia have had a lesion in a sense of their connective, but one expresses this increase. And what's interesting is this G3 will be our lead ganglion, all right? So we're starting to get insights into the cell molecular changes that are occurring across this transection that may help us explain what's changing in the lead ganglion, in addition to this ingrowth of these proprioceptive cells. And Dave has gone on to look at changes in ion channel expression. Here's the transection between ganglion 2 and 3. And for example, he sees lots of upregulation in ion channel expression. Here's SHAB1, and downregulation in the ganglion adjacent to the transection. So we have definitely many more questions than answers, but we think it's a phenomenal preparation in which you can study how it's centrally driven locomotor program can switch to one that is peripherally controlled. We know in the field of locomotion that there's some preparations that are heavily dependent on the periphery for the timing of its intersegmental coordination. And then we know we have like Ansgar's stick insects, very heavily reliant upon sensory information. And then with other systems that are very centrally driven. And because we get really beautiful intersegmental crawling and their coordination with the leech in the dish, we know that everything's fine with the central pathways. So basically by studying the lead ganglia in particular and the remodeled stretch receptors, we hope to address this important issue in the field of motor control, especially as we travel down the path of understanding action selection in the context of homeostatic plasticity. And I just want to thank a few folks here. This is an older photo. I don't know folks that know Mara, but Mara was in my lab for a while before she went to work with Eve, and she helped with the recovery project. And here's Josh Poole and Cindy Harley, who helped with the recovery project. And Tony was our undergrad who could fill individual stretch receptors. He's now a grad student at the University of Washington, and especially my collaborator, Dave Schultz. And with that, I'm open to questions.
Darren, can I ask? So you showed crawling that looks perfectly coordinated with the sucking of the front and back suction apparatus. How are those coordinated if it's fully transected?
That's a great question, Jorge. And I have a pet hypothesis that for the sucker information, maybe afferent information is also somehow growing through the body into somehow getting into the nerve roots.
That's very
right?
long
Because
distance
the
growth.
peripheral nervous system, any kind of proprioceptive input, seems to want to get back into the CNS. Right? So that may be one answer to that question, but you know, it's intriguing how, I mean, it's a whole nother layer to the story is how the suckers are somehow being
Stuck into the
stuck back onto the rhythm that the leech has. I mean, there's plenty of work to be done in this system.
Nice, thank you.
Karen, what is the sensor for water depth?
I don't know what that sensor is. I really don't, and I've thought about it for years. I mean, we have touch and pressure sensitive cells, and I mean, what we know about them, I can't imagine there's that much pressure on the underside of the leech that, I mean, it could be that the pressure sensitive cells per se help to gate information with that decision to swim or crawl.
What about vision?
You know, so John Jellies had found, yes, that vision is important in knowing whether the leech is right side up or downside, but there's not many changes in the visual aspect, Mikey, for when the leech's body is just floating and then all of a sudden in contact with the substrate, which is lit up. It's Silgard. We got a lot of a dark field. We're using dark field microscopy, so we're shooting up a lot of light so we can see the ganglia recording from. So the bottom of the dish isn't dark, it's actually light. So I
Actually,
think
yeah,
the
future.
vision, I mean, I thought about the vision, but I don't think that could explain it, but I'm willing to test it.
I have another question as well, and that is, do you have any way to record extracellularly from the nerve cord for RB3? Because it's possible, as you know, these neurons survive after transection for months, if not longer,
Right,
but may still be driving all the business.
right. So I guess I have two answers. So one is that, we took away the brain. So the idea would be the axon of R3B1 was still viable.
Right.
And I know how to record from the connective when I have the cell body recording. If I stimulate the connective by itself, I cannot see a distinct unit that I could identify as R3B1 because there's just too many units.
But if you have two recordings, one in the front and one in the back.
Right. I haven't been able to, we tried this many years ago and there just were too many units. I couldn't really see anything that stood out. But I hear what you're saying. As we did exactly that experiment, we could see the spike in the cell body of R3B1, and we could set the timing and look for it in the posterior ganglia, but we just weren't, I don't know, there's just too many units that I couldn't pick out when I go posterior to the brain, because there's a bunch of other cells talking to each other in the mid-body ganglia. But there is an experiment I would like to do, and that is we had gotten some of our voltage sensitive dyes to work, and we could drive crawling, right, with R3B1. What I'd really like to do is see if I can drive crawling by stimulating a stretch receptor or combination of stretch receptors. And I don't think R3B1 is surviving. And the reason is, Mikey, we have had some leeches that are now like over a year old and more. And I don't think R3B1's axon would survive longer than 12 months. Right? And these are animals that, they only feed twice a year, so they're surviving and they look great. And they have super good cross-specific intersegmental coordination at like 14 months. And I just can't believe R3B1 would still be alive.
Now, you're a biologist. You know better than to not believe something.
Well, I think it would be kind of cool. Then my question is, why can't I get it in the dish? If the axon of R3B1 is alive and well, then I should put dopamine on the whole cord and R3B1 should do its thing, and it doesn't. You need the periphery. So why do you need the periphery if the axon of R3B1 is just fine?
Maybe it's the periphery that's activating R3B1.
R3B1? Well, that's possible. Anything's possible, Mikey.
All right, I'll wait for your next paper.
Karen,
Yeah,
I was wondering about the swimming network after you remove the descending input and you release it from that inhibition. What then are the signals that are controlling whether it's active or quiet?
I have no idea. My thinking is it has to do with these proprioceptors.
Mhm.
And what I'd like to find out is the timing of the ingrowth of these proprioceptors. And it seems like when the regrowth of the terminals of these proprioceptors basically project to an anterior or a posterior adjacent ganglion. Right? I mean, we think it's about the same time that we get the onset of crawl coordination, but the suppression of swimming. Otto has shown these proprioceptors play a role in swimming. And so maybe this new regrowth somehow also helps to suppress the swim oscillators in some way.
Okay. I love those stretch receptors growing past where
Yes,
they normally stop.
And
Okay.
having worked on Manduka, where you have this wonderful remodeling of a nervous system to generate the moth from the caterpillar, remodeling is very near and dear to my heart. And you don't remodel for no reason. Remodeling plays an important role. It's telling us that now we have stretch receptors that have never, ever had any direct intersegmental association with other cells. I mean, and so they're ignoring the boundaries of their ganglion, the chemical cues that prevented them from growing across to other ganglia. And now they're growing in a way, forming potentially maybe a ladder-like structure to help with the intersegmental coordination. I think it'll be really important to see if we can entrain the rhythm by moving the stretch receptors back and forth. And I think for robotic system, Joe, to go from a system that is really almost entirely centrally driven to potentially entirely peripherally driven is a really cool system. How that works. Because we no longer have a fictive prep anymore. It doesn't work in the dish. There are no central elements sufficient to drive coordinated, crawl-specific coordinated behavior. That's lost. So when we take nervous systems from leeches that are about a year old and put them in the dish and put on dopamine, they will not show that intersegmental coordination. But they move beautifully, you know, in the aquarium. Guys, have any more questions?
Thank you, Karen. I'm going to stop the recording now.
Thanks, Karen. That was
Thank you,
super
Karen.
cool.
That was nice.